Human gut microbiome is an intricate ecosystem composed of trillions of microorganisms, including bacteria, viruses, and fungi that play a vital role in maintaining overall health. Among these, certain strains of bacteria have been found to exert a profound influence on the body's aging process, particularly at the cellular level. These bacteria are not merely passive residents of the digestive tract. They are active biochemical participants that engage with the immune system, regulate inflammation, and influence the production of key neurotransmitters and hormones. Research has increasingly shown that the composition and diversity of gut bacteria can either support or sabotage the body's efforts to maintain vitality and energy over time. When the microbiome is rich in beneficial bacteria, such as Akkermansia mucinifera or certain bifidobacteria, it fosters an environment where inflammation is reduced, metabolism is regulated efficiently, and cellular functions operate more effectively. These effects extend to the mitochondria, the energy-producing organelles within our cells, which are essential for sustaining physical energy, mental clarity, and overall longevity. As the body ages, mitochondrial function tends to decline, leading to reduced energy levels and an increased susceptibility to disease. However, emerging evidence suggests that specific bacterial strains in the gut can directly support mitochondrial health by producing metabolites such as short-chain fatty acids, SCFAs, including butyrate, acetate, and propionate. These compounds act as signaling molecules that influence gene expression related to cellular repair, anti-inflammatory responses, and oxidative stress reduction. Moreover, certain anti-aging pathways like AMPK activation and sirtuin regulation appear to be modulated by the gut microbiome, indicating a profound link between microbial presence and the molecular mechanisms that govern aging. When beneficial bacteria are plentiful, they help maintain the integrity of the gut lining, preventing a condition known as leaky gut that can allow toxins and pathogens to enter the bloodstream, triggering chronic systemic inflammation. This low-grade inflammation, often unnoticed, accelerates cellular aging and disrupts the balance of hormones that regulate mood, metabolism, and immune responses. Furthermore, these bacteria influence the vagus nerve, a primary conduit between the brain and gut, thereby affecting the body's stress response and cognitive function. The brain-gut axis is now recognized as a critical pathway through which microbial activity can shape mood, memory, and even neuroplasticity. By optimizing the microbiome through diet, lifestyle changes, and possibly targeted supplementation, individuals may enhance not only their physical energy, but also their psychological resilience and cognitive longevity. Fermented foods, prebiotics, and polyphenol-rich diets have been shown to encourage the growth of these beneficial microbes, which in turn produce enzymes and cofactors necessary for maintaining cellular youth. What's particularly groundbreaking is the understanding that these bacteria are not static. They respond dynamically to our behaviors, stress levels, and environmental exposures. This means the process of aging is, to a large extent, malleable through the gut. With ongoing research, scientists are now exploring microbiome transplantation, engineered probiotics, and precision nutrition as potential strategies to harness these bacteria for anti-aging benefits. A balanced microbiome plays a central role in slowing biological aging and supporting hormonal health, but acting as a regulatory hub for many of the body's essential systems. Within the gut are trillions of microorganisms that not only help digest food, but also influence systemic functions such as immune modulation, inflammation control, and the production of neurotransmitters and hormones. These microbial communities are highly sensitive to diet, environment, and stress, and when balanced, they create a symbiotic relationship with the human host that favors longevity and vitality. One of the key mechanisms by which a balanced microbiome supports slower aging is through the regulation of systemic inflammation. Chronic low-grade inflammation, often called inflammaging, is a major contributor to age-related decline. A healthy and diverse gut flora produces metabolites such as short-chain fatty acids that help modulate immune responses, suppress inflammation, and promote tissue repair and regeneration. This anti-inflammatory environment is essential for maintaining hormonal stability. Hormones like cortisol, estrogen, insulin, and thyroid hormones are deeply influenced by gut bacteria through multiple pathways. The microbiome can influence cortisol levels via the hypothalamic pituitary adrenal HPA axis, and its effect on estrogen metabolism is especially significant in women's health. Certain bacteria known as the estrobolome play a role in breaking down and recycling estrogen within the body, thereby impacting mood, reproductive function, skin health, and bone density. When the microbiome becomes imbalanced due to factors like antibiotics, poor diet, or chronic stress, the estrobolome is disrupted, which can lead to estrogen dominance or deficiency, both of which accelerate aging symptoms and increase the risk of disease. Similarly, insulin sensitivity and glucose metabolism are regulated in part by the gut microbiome, with certain bacterial strains aiding in blood sugar regulation and reducing the risk of metabolic disorders such as type 2 diabetes. The gut microbiome also communicates with the endocrine system to modulate the release of neurotransmitters such as serotonin and dopamine, which are critical not just for emotional well-being, but also for sleep, motivation, and the body's ability to recover and regenerate. Approximately 90% of the body's serotonin is produced in the gut, showing a direct link between microbial health and mood, energy levels, and cognitive performance. A balanced gut ecosystem supports better sleep quality by influencing melatonin production through serotonin regulation, and this in turn enhances the body's ability to repair itself overnight when many anti-aging processes such as autophagy and cellular detoxification are most active. In terms of nutrient absorption, a well-functioning microbiome ensures efficient digestion and extraction of critical micronutrients like B vitamins, magnesium, zinc, and vitamin K2, which are essential for mitochondrial energy production, DNA repair, and hormonal synthesis. Without these nutrients, the body's repair mechanisms become compromised, leading to faster cellular degradation and visible signs of aging. The microbiome essentially acts as a factory for bioactive compounds and cofactors needed for the body's natural longevity pathways to function optimally. A disruption in microbial balance can tip the body into a state of hormonal chaos and metabolic dysfunction that accelerates aging from the inside out. Mitochondrial function, the process by which cells produce energy, is deeply intertwined with microbial activity in the gut. Mitochondria are the energy generators of the body, converting nutrients into adenosine triphosphate, ATP, the fuel that powers every cellular process. Their performance determines how energetic, resilient, and youthful a person feels. Recent scientific discoveries have revealed a compelling relationship between these cellular powerhouses and the microorganisms that reside in the gut. This relationship is both ancient and complex, rooted in evolutionary biology, as mitochondria themselves are believed to have evolved from bacteria through a process called endosymbiosis. Because of this shared origin, mitochondria and gut microbes communicate through biochemical signals that influence each other's function, and this interaction has profound implications for aging and energy regulation. Gut bacteria produce a wide variety of metabolites that directly affect mitochondrial health. Among the most important are short-chain fatty acids like butyrate, acetate, and propionate, which not only serve as fuel for cells lining the colon, but also play key roles in reducing oxidative stress within mitochondria. Oxidative stress, caused by an imbalance between reactive oxygen species, ROS, and the body's ability to detoxify them, is one of the primary drivers of cellular aging. When ROS accumulate unchecked, they damage mitochondrial DNA, reduce ATP production, and initiate programmed cell death or senescence. Beneficial bacteria help buffer this oxidative load by enhancing the expression of antioxidant enzymes and promoting mitochondrial biogenesis, the process by which new mitochondria are formed in cells. This rejuvenation of mitochondria is critical for sustaining energy levels and maintaining healthy tissues and organs. The gut microbiome also influences mitochondrial function through its regulation of systemic inflammation. Inflammatory cytokines can impair mitochondrial respiration and cause mitochondrial fragmentation, a process associated with fatigue, muscle weakness, and cognitive decline. Conversely, when gut microbes are in balance, they help keep inflammation in check, which preserves mitochondrial integrity and efficiency. The link between gut health and mitochondrial activity also extends to the regulation of insulin sensitivity and glucose metabolism. Certain bacterial strains help maintain stable blood sugar levels, reducing the risk of insulin resistance, a condition that damages mitochondria and accelerates aging at the cellular level. Furthermore, specific gut-derived metabolites activate key longevity pathways such as AMPK and SIRT1, both of which are involved in enhancing mitochondrial function and extending cellular lifespan. These pathways also regulate autophagy, a cellular process that removes damaged mitochondria and recycles their components to build newer, more efficient energy producers. This cleanup process is essential for sustaining high-performance energy metabolism and delaying the onset of age-related diseases. Diets rich in prebiotics, fiber, and polyphenols support the growth of gut bacteria that stimulate these pathways, creating a feedback loop in which better gut health leads to better mitochondrial health and vice versa. The nervous system also plays a role in this interaction, particularly through the gut-brain axis. Signals from the brain influence gut motility and microbial diversity, while microbial metabolites can cross the blood-brain barrier and affect brain mitochondria. This neuromitochondrial axis is especially important in maintaining cognitive energy and preventing neurodegenerative disorders. When mitochondrial function is compromised, energy-intensive organs like the brain and heart suffer first, leading to symptoms often attributed to aging chronic inflammation that results from poor gut health is one of the most powerful accelerators of aging and cognitive decline. Inflammation, when controlled and temporary, is a vital part of the body's immune defense system. However, when it becomes persistent and systemic, it turns into a silent killer that damages tissues, impairs cellular communication, and disrupts essential biological functions. The gut is a primary interface between the outside world and the body's internal environment, and it houses more than 70% of the immune system. When the gut lining is damaged or when harmful bacteria outnumber beneficial ones, it creates a condition often referred to as dysbiosis. Dysbiosis leads to increased intestinal permeability, sometimes called leaky gut, where microscopic particles such as toxins, undigested food proteins, and bacterial byproducts escape into the bloodstream and trigger a widespread immune response. This continuous low-grade immune activation causes the release of pro-inflammatory cytokines, which circulate throughout the body and disrupt systems far beyond the gut. Over time, this chronic inflammation becomes a central driver of biological aging, contributing to the breakdown of tissues, the shortening of telomeres, and the decline in cellular repair mechanisms. It also negatively impacts mitochondrial function, reducing the production of ATP and increasing the release of reactive oxygen species, which further damages cells and accelerates age-related degeneration. This self-reinforcing cycle, in which inflammation impairs energy production and damaged cells create more inflammation, becomes harder to interrupt as we age. The brain is one of the organs most vulnerable to inflammation driven by gut dysfunction. Inflammatory molecules that cross the blood-brain barrier can alter the function of neurons, impair neurotransmitter production, and reduce synaptic plasticity. This leads to brain fog, memory problems, mood disorders, and a greater risk of neurodegenerative diseases such as Alzheimer's and Parkinson's. The gut-brain axis, a complex communication network between the central nervous system and the enteric nervous system, plays a crucial role in maintaining cognitive resilience. When this axis is disrupted by inflammation, it can also disrupt sleep, mood regulation, and emotional stability, all of which are essential for healthy aging. A poor diet high in processed foods, sugar, and artificial additives contributes significantly to gut inflammation by feeding harmful bacteria and starving beneficial microbes of the fiber and nutrients they need to thrive. This microbial imbalance produces endotoxins like lipopolysaccharides, LPS, which have been shown to trigger strong inflammatory reactions and contribute to the development of insulin resistance, obesity, and cardiovascular disease. These chronic diseases are not just common in aging populations, but are also strongly linked to accelerated cognitive decline. Additionally, stress, lack of sleep, and environmental toxins all add to the inflammatory burden by further destabilizing the gut microbiome and weakening the immune system's ability to respond appropriately. Restoring gut health through nutrition, stress management, and microbial diversity can help reduce this chronic inflammation and slow down the biological processes that lead to aging. A diverse and balanced microbiome acts like a firewall, protecting the body from inflammatory triggers and maintaining homeostasis across multiple systems. Fermented foods, prebiotics, omega-3 fatty acids, and regular physical activity have all been shown to reduce gut inflammation and support long-term cognitive and physical health. Intermittent fasting and specific probiotics found to enhance longevity pathways by interacting directly with cellular processes that regulate aging, inflammation, metabolism, and repair. Intermittent fasting, which involves cycling between periods of eating and fasting, has been shown to activate crucial longevity, promoting mechanisms such as autophagy, mitochondrial biogenesis, AMPK activation, and sirtuin regulation. Autophagy is the body's internal recycling program, where damaged or dysfunctional cellular components are broken down and reused to build new, healthier structures. When autophagy is triggered through fasting, the body begins to clean out old or malfunctioning cells, particularly those contributing to aging and disease. This cellular housekeeping process not only extends the lifespan of individual cells, but also enhances the functionality of tissues and organs across the body. AMPK, an energy sensing enzyme, is another pathway stimulated by intermittent fasting. When activated, AMPK increases insulin sensitivity, supports fat metabolism, and improves mitochondrial function, which in turn boosts energy efficiency at the cellular level. Sirtuins, a family of proteins involved in gene expression and DNA repair, are also activated by fasting and are directly linked to enhanced longevity and stress resistance. These cellular pathways collectively create a state of enhanced repair and maintenance, reducing the buildup of cellular damage that is typically associated with aging. Intermittent fasting also helps regulate insulin levels and reduce oxidative stress, which contributes to a more youthful internal environment and lower risk for metabolic disorders. At the same time, specific strains of probiotics, live beneficial bacteria introduced into the gut, play a complementary role by maintaining microbiome balance, improving gut barrier function, and producing metabolites that influence systemic health. Some of the most researched longevity supporting probiotics include Lactobacillus planarum, Bifidobacterium longum, and Akkermansia mucinifila. These strains are known for their ability to reduce inflammation, enhance gut integrity, and produce short-chain fatty acids that benefit the brain, heart, and immune system. These SCFAs not only act as anti-inflammatory agents, but also serve as energy sources for colon cells and signaling molecules that influence gene expression in organs far beyond the digestive tract. When intermittent fasting is paired with the right probiotic support, the synergistic effect creates a favorable internal environment for anti-aging at both the cellular and systemic level. The gut becomes more resilient, inflammation is reduced, and the communication between the gut and brain becomes more efficient. Probiotics can also improve the body's absorption of key nutrients required for energy metabolism, such as B vitamins, magnesium, and zinc, which support mitochondrial health and cellular regeneration. In addition, certain probiotics have been found to modulate neurotransmitters like serotonin and dopamine, contributing to improved mood, sleep quality, and cognitive clarity, all of which are critical factors in extending not just lifespan, but also health span. By supporting the microbiome through these targeted approaches, the body becomes better equipped to handle oxidative stress, detoxify harmful substances, and regenerate tissues more efficiently. These practices reinforce each other by influencing hormonal balance, stabilizing blood sugar levels, and promoting metabolic flexibility, which is the body's ability to switch between fuel sources like fat and glucose, depending on energy demands. Sleep, sunlight, and microbiome diversity are non-negotiable factors for vibrant aging because they serve as fundamental pillars that regulate circadian rhythms, immune balance, energy metabolism, and cellular repair. Each of these elements independently contributes to longevity, but when optimized together, they form a powerful synergy that enhances physical vitality, cognitive clarity, and emotional resilience as the body ages. Sleep is a biologically essential process where the body enters a state of restoration and detoxification. During deep sleep, the brain undergoes glymphatic clearance, where waste products such as beta amyloid are flushed out, reducing the risk of neurodegenerative diseases like Alzheimer's. Simultaneously, Sleep activates the release of growth hormone and promotes autophagy, processes crucial for repairing tissues, strengthening immunity, and maintaining the integrity of cellular structures. Lack of sleep, on the other hand, increases levels of cortisol, disrupts insulin sensitivity, and leads to chronic inflammation, all of which are hallmarks of accelerated aging. Sunlight, particularly early morning sunlight, plays an essential role in synchronizing the circadian clock, which governs sleep, wake cycles, hormone secretion, digestion, and metabolism. Exposure to natural light stimulates the production of serotonin, a neurotransmitter that boosts mood and is a precursor to melatonin, the hormone responsible for sleep regulation. When circadian rhythms are aligned through consistent exposure to natural light, the body experiences more restorative sleep, balanced hormone levels, and improved cellular efficiency. Sunlight also enables the synthesis of vitamin D, a hormone like vitamin that is vital for immune function, bone health, and the prevention of age-related conditions such as cardiovascular disease and osteoporosis. Deficiency in vitamin D has been linked to fatigue, cognitive decline, and reduced immune resilience, making sunlight a natural and powerful longevity tool. Microbiome diversity, the presence of a wide range of beneficial bacterial species in the gut, acts as a foundational mechanism for vibrant aging. A diverse gut microbiome produces a broad spectrum of metabolites that influence not just digestion, but also immune modulation, inflammation control, and neurotransmitter production. High microbial diversity is associated with stronger gut barrier function, which prevents toxins and pathogens from leaking into the bloodstream and triggering systemic inflammation. It also supports nutrient absorption, particularly of compounds like magnesium, iron, and B vitamins that are necessary for mitochondrial function and energy production. The presence of many different strains of bacteria ensures that the gut can adapt to various challenges, such as dietary changes, infections, or stress, without losing its protective functions. Together, sleep, sunlight, and microbiome diversity create a feedback loop that sustains vitality and delays the onset of age-related degeneration. Quality sleep enhances microbial balance and immune health, while a healthy gut microbiome in turn improves sleep quality through the production of neurotransmitters and hormones. Sunlight supports both sleep and microbial diversity by regulating circadian rhythms and encouraging behaviors such as physical activity and outdoor exposure, which are linked to a healthier gut. These three factors are not just lifestyle choices, but biological necessities that influence how the body regenerates, adapts, and thrives through the aging process,